Okay, as you know, we're running a little bit behind schedule, so we're going to move right into the next presenting company, and that is Sangamo BioSciences. Presenting for them today, this is her second time up here today, is Elizabeth Wolfe. She is the VP of Corporate Communications. Elizabeth.
Okay, let me get this straight. So I've only got 14 minutes. First of all, thank you very much to the Alliance for Regenerative Medicine for including Sangamo in this conference today. I will be making forward-looking statements, so I'd refer you to the documents that we file with the SEC. What I'd like to do today is to give you a rough idea of or to outline for you the technology that Sangamo is developing, the kinds of therapeutic applications that we're going for, our recent progress, and outline our upcoming milestones. So Sangamo is, let me introduce Sangamo to you and give you an idea of the therapeutic directions that we're taking. We're developing a powerful technology, our zinc finger DNA binding technology, or ZFP technology, which, as the name suggests, functions at the DNA level. Now, the ability to function at the DNA level gives us a range of capabilities that aren't available with conventional therapeutics. And it means that we can begin to think about actually making a real possibility, the possibility of engineering genetic cures for diseases. We're focused, obviously, in genetic diseases, and Sangamo is particularly focused in monogenic diseases. So we have been developing this technology for a number of years. It's really been the entire focus of the company. We're able to do both targeted genome editing and targeted gene regulation with high precision. We've also developed broad capabilities in delivery, and have used the technology to develop broad commercial applications in a variety of different, with a variety of different companies. These strong partnerships have meant that we can maintain a strong balance sheet, and we have a dominant intellectual property position. So the earliest programs that the earliest partnerships that we established were in non-therapeutic applications of the technology with Dow AgroSciences and Sigma Aldrich in plant agriculture and research tools. More recently, we've begun collaborations with in therapeutic areas. Back in 2012 with Shire in hemophilia A and B, and Huntington's disease, and most recently with Biogen IDEC in the hemoglobinopathies. But these are limited licenses, and Sangamo retains broad rights to the platform, and we're developing a number of monogenic disease applications of our own. So let me tell you a little bit about the technology. We begin with a zinc finger DNA binding protein. These are natural proteins. They're present in organisms from yeast through to man. They've evolved with our complex genomes. This very time, there are hundreds of zinc finger proteins doing their DNA binding thing in you and me. And we can engineer these proteins to bind to any sequence that we desire. So any sequence in any cell type, in any genome. We can also make sure that they only bind to the sequence we want. And we've been working on this for a long time. We're very good at it. We can add functional domains to this zinc finger moiety in order to achieve a number of different outcomes. We can achieve gene regulation by adding an activation domain. We can activate the expression of genes and increase the level of protein expression. We can also repress genes, and this is what we're doing in our Huntington's program, as you'll hear later with Shire. The other aspect of the technology that we're developing is this nuclease technology, which enables us to do very precise genome editing. And by adding a nuclease domain to a zinc finger protein, we can use the cell's machine, we can make a double-strand break in the genome, and then use the cell's machinery to affect a number of different outcomes. We can knock out a gene, as we're doing with our HIV program, where we're knocking out CCR5, or we can correct or add a particular gene sequence into a defined location in the genome. This could be a couple of base pairs or an entire gene coding sequence. So let me give you a little taste of what we're doing with Biogen IDEC. We're focused there in the hemoglobinopathies focused on sickle cell disease and beta thalassemia. These are two monogenic diseases which are both caused by mutations in the adult hemoglobin gene. Now, we know that, in fact, individuals with either sickle cell or beta thalassemia actually have a fully functional globin gene in their genome. That's because babies born with this disease don't exhibit symptoms in the early stages. Those symptoms only come on later, as they develop through infancy. And the reason for that is that in utero and in early development, individuals express fetal hemoglobin, which is a fully functional form of globin, and it's only when, as they go through development, that the adult globin is switched on. We also know that individuals that continue to express who have got beta thalassemia or sickle cell disease and continue to express fetal hemoglobin in later life have milder symptoms of disease. So the experiment has been done for us. What we're aiming to do is to maintain the expression of fetal hemoglobin. And this we're doing by going after one of the master switches of this transition from fetal to adult hemoglobin, the BCL11A gene. And what we've done is to make zinc finger nucleases that enable us to knock out BCL11A very specifically in hematopoietic stem cells from individuals with beta thal or sickle cell disease. And we're developing this as a cure for both diseases. So we recently presented an update from this program at the American Society for Hematology meeting, or ASH, in December, where we showed the feasibility of this approach in hematopoietic stem cells, and we also presented the clinical manufacturing, just demonstrating that we can efficiently manufacture this at clinical scale. Let me move on to our programs with Shire. This is another application of the technology, but this time in an in vivo setting. What we're looking to do is deliver zinc finger nucleases in vivo, and in this time to do gene addition. So we're focused here on hemophilia with Shire, but this same technology can be used for, and Sangamo is taking it forward in lysosomal storage disorders. So what we do is the following, and the way that we've been developing this is we wanted to develop a system that could be used broadly across a number of diseases which are currently being treated by enzyme replacement therapies. And so we picked a gene that's only expressed in the liver, that's expressed at enormous quantities, and this is the albumin gene. And we know that you can actually co-opt a small amount of this gene in order to express a gene, a foreign transgene. And we've shown this. So what we do is we start with our zinc finger nucleases, and I don't think my red spot will reach as far as there, but you start with zinc finger nucleases. They're designed to bind just downstream of the albumin promoter and make a double-strand break. And what we do then is introduce into the cells what we call a donor DNA, which contains our gene of interest. This, we let the cell's repair machinery do its thing, and what happens is that the gene of interest is specifically and very precisely loaded in behind the albumin promoter and runs off the albumin promoter. With Shire, we're using this for hemophilia A and B, but this can be used for any other gene that you want. We recently updated on these programs at the American Society for Hematology, and we showed that albumin supports, the albumin locus supports robust expression of therapeutic transgenes. In mouse models of hemophilia A and B, we demonstrated that we can get therapeutic levels of protein expressed that correct the defect. We've moved into larger animals, into non-human primates, and show that we can get modification of the albumin locus in rhesus monkeys, and that we can get expression of therapeutic levels of protein from this locus. As I said, if successful, this can be leveraged across a number of different indications, and Sangamo is pursuing lysosomal storage disorders. So I want to move to another aspect of the technology. This is an in vivo direct application in the brain, but using our gene regulation technology, and this is a program we're taking forward with Shire. So Huntington's disease is a fatal neurodegenerative monogenic disease, again caused by a mutation in a single gene. And the gene is kind of interesting in that it's an expansion of a region of the gene that's present in the normal allele. So normally you would have, and the expansion is what they call a CAG repeat, so a repetitive sequence of DNA. Normally you would have around 20 repeats of this. In the disease allele, you have greater than 35. You only need one mutant allele in order to manifest the symptoms of disease sometime in your life. And so what we're looking to do, the one other thing I should mention is that we know that production of the normal gene is required. So this protein is an essential function. And so anything that's going to address the Huntington's disease paradigm is going to need to be able to repress the mutant form whilst leaving the expression of the normal form alone. And we've designed zinc finger transcription factors that selectively repress only the mutant form and leave the normal form untouched. We recently updated at the Society for Neuroscience meeting in November of last year and showed in both human patient-derived cells and mouse models that we consistently successfully repress expression of mutant disease-causing Huntington to levels of up to 90%, whilst leaving the wild-type gene unchanged. We also looked in mouse models for both chemical and physical signs of efficacy, and we could see that we saw a reduction in aggregates of mutant Huntington protein in ZFP-treated regions of the brain, increased levels of protection of a neuronal cell type that degenerates in Huntington's disease patients, and importantly, statistically significant reduction in clasping behavior. And this is a behavior in the mouse that mimics motor symptoms of Huntington's disease. So we're using in vivo delivery, using AAV directly into the brain for this indication. Finally, I'm going to mention very, very briefly our HIV/AIDS program. This is an ex vivo approach in T cells where we're knocking out a gene, the CCR5 receptor. The phase 1 data for this study were recently published in the New England Journal of Medicine. These were the results from a phase 1 study that was carried out at the University of Pennsylvania. It was the first in-man study of genome editing. And the data essentially showed that the treatment is generally safe and well tolerated. We see a marked increase in CD4 cells. These cells engraft, traffic, persist. We also know that these cells have a selective advantage over unmodified cells when exposed to the virus, and that was shown in the paper. And there was a key subject reported in this paper who managed to control his viral load to levels at or below the levels of detection whilst off ART for a treatment interruption, and managed to control those went to undetectable levels before going back onto ART. So that was a phase 1 study. We've actually moved on quite a long way from there. We've treated over 70 patients in a variety of different protocols. We're currently in a phase 2 study, and this slide just summarizes the data from those studies. What I want to point out is that we have seen a number of instances where patients can control their viral load down to undetectable levels. And in fact, one patient that we have in our most recent study is out now over 7 months without antiretrovirals after a single treatment with Sangamo's modified cells. And then the other thing that I'd like to point out is that we're seeing reduction in the HIV reservoir, which is an area that the antiretrovirals don't touch. So one of the reasons when you come off antiretrovirals that the virus comes roaring back is because there's a significant reservoir of HIV in T cells. And so what we've seen is that in 9 of 9 subjects, and these were subjects that had had HIV for a long time and had fairly low CD4 counts when they came into the trial, in 9 of these subjects, we've seen a reduction in their viral reservoir over a space of 3 years after a single treatment with SB728T, which is our modified cells. So next steps, we're involved in, we're carrying out a 12-patient phase 2 study to further explore this, looking at both optimal levels of a conditioning protocol to increase the levels of modified cells, and at inclusion criteria based on some of the things we've learned in previous trials. And one other thing that we'll be implementing in these protocols is mRNA delivery, which we believe will not only be more efficient than the current delivery protocol using adenovirus, but will allow us to retreat. And just to give you an idea of our upcoming milestones, we've got a lot ahead of us in the next year. We're expecting to complete enrollment in our HIV T-cell study in 2014. We will also initiate a stem cell study, and we'll file the IND in 2014 for a stem cell study. This is using the same zinc finger nucleases that we've used in the T-cell program. We'll be filing INDs for hemophilia A and B with Shire and beta thalassemia in 2014 with Biogen IDEC. And in 2015, have another 4 INDs in sickle cell, 2 LSDs, which Sangamo is taking forward, and we'll give more details on that later in the year, and also Huntington's with Shire. And last but not least, we have a Serogene 110. This is a program that we acquired from Serogene when we acquired it back in October of last year, and those data will read out in 2015. So I'm going to summarize by saying I hope that you take away from this that we have a robust technology. We're focused largely on monogenic diseases and infectious diseases with the aim of not just providing a new treatment paradigm that gives you a longer-lasting drug, but actually engineering genetic cures, a permanent solution to some of these problems. We have multiple INDs coming through in the next, that we'll be filing in the next couple of years, 2 phase 2 data readouts. And we have multiple partnerships that are helping us take these programs forward to points of value inflection, and a strong balance sheet in order to be able to carry forward this therapeutic pipeline. And we've tried very hard when we've been thinking about how we approach the technology and how we capitalize on it to make sure that we're taking things forward, both proprietary programs and partner programs. We're looking at different ways of delivering the ZFPs and using all the different modalities of it, and looking very hard at the targets that we choose, looking to make sure that we have a target where a single gene is associated with a disease, monogenic diseases, and so our technology, the use of our technology makes a lot of sense. And finally, we have the balance sheet strength to carry it forward. Thank you very much for your attention.